Inside almost every single cell in your body, there is a sequence of 3 billion chemical letters, a massive, twisting repository of genetic information. A sequence of 3 billion letters remains uninformative until a computational system can distinguish meaningful signals from the surrounding data. Without a map to identify where critical elements begin and end, the raw sequence provides very little insight. In June of 2000, world leaders gathered to announce the completion of the Human Genome Project, a milestone often described as decoding the book of life. Sequencing the human genome provided the raw data. The subsequent challenge lay in parsing that information into a functional map of human biology. This is where genomics takes over. While genetics focuses on how specific traits are inherited, genomics uses computational power to study an organism's entire genetic landscape all at once. To make sense of this data, bioinformaticians analyze the genome across 3 distinct scales of resolution. We look at the structural, the functional, and the comparative dimensions. Making sense of the genetic code requires constantly zooming in and out across these layers, connecting the physical map to real-time activity and deep evolutionary history. The first scale is structural genomics, the physical mapping phase. Before we can determine what a gene does, we need to know its precise location on the chromosome, along with the regulatory elements that control its behavior. This is a massive sorting task. Only a tiny fraction of human DNA actually contains the code to build proteins, meaning researchers must carefully separate coding sequences from vast regions of non-coding and repetitive elements. Researchers use the UCSC genome browser to visually organize stacked layers of annotation tracks, like gene predictions and mRNA sequences. By toggling data tracks, they isolate critical features, using menus to identify individual exons or hide introns to pinpoint phenotype associations. Structural genomics transforms a linear string of text into a highly organized, navigable physical map. Once we have a map, we must move to the second scale, functional genomics. We need to see the genome in action. This is the realm of the transcriptome, the complete set of RNA transtrants produced by the genome. Genes operate as part of a dynamic network, switching on or off in response to cellular stress, environmental changes, or disease. Using tools like microarrays or RNA sequencing, scientists measure this activity globally. Instead of looking at one gene at a time, they track thousands of genes simultaneously, seeing which ones are upregulated and glowing bright, and which are downregulated and dim. This interconnected activity functions as a reactive operating system, constantly shifting its state in response to the organism's environment. Finally, we pull all the way back to the widest scale, comparative genomics. Here, bioinformaticians compare the complete genetic codes of entirely different species. This relies on the core principle of evolutionary conservation. If a specific genetic sequence remains unchanged across millions of years of evolution, it likely performs a biologically essential function. Sequence alignment algorithms are designed to hunt for these exact patterns. By stacking human DNA against model organisms like mice or chimpanzees, the algorithms identify highly conserved regions, illuminating the columns that match across species. By mapping the differences and similarities between species, we identify the universal biological mechanisms that define all known life. Combine the physical map, the dynamic network, and the evolutionary history, and you get actionable medical science. In the field of pharmacogenomics, doctors identify specific genetic mutations in a patient, and then they use that data to design targeted drug therapies that lock perfectly into a specific cellular receptor. This approach extends beyond human medicine. We can engineer resilient agricultural crops and track global outbreaks in real time. Genomics bridges the gap between raw biological data and life-saving engineering. All of this analysis relies on a massive digital architecture, open-source databases like NCBI and Ensemble that freely host bioinformatics data for the global scientific community. Genetic instructions eventually translate into physical work, carried out by the diverse population of proteins that build and maintain our cells. This brings us to the next necessary step in bioinformatics, proteomics. To understand how biology functions, researchers must map the 3D structures these genetic instructions build. This image shows a structural comparison of myoglobin and hemoglobin. Descended from a common ancestor, they share highly similar structures despite differing sequences. Notice how the blue and pink ribbons of myoglobin and beta globin superimpose perfectly, demonstrating their structural homology. Genomics maps the code of life, but to understand the physical machines that execute that code, we must enter the world of proteomics. We'll explore exactly how that works in our next video.